Okay, so we've been working on blood flow restricted exercise for, I would say, 15 years since I started here at Loughborough, and it's been a sort of interesting journey so far. In terms of where we've come from, we started with, the use of blood flow restricted exercise with resistance exercise and resistance training, but it really became apparent that, the interest in endurance performance is novel and potentially unique. So in terms of using blood flow restricted exercise, before we start that, I just want to sort of give you a very, very brief overview of the main determinants of endurance performance. And this is very much plagiarized from the classic Joyner and Coyle model from 2008. We've modified it slightly, but clearly endurance performance is determined by the aerobic contribution to exercise, which is underpinned by VO2 max and the various threshold parameters. We won't get into a debate about those specific ones, but we're talking about lactate threshold and critical power. And then the role of the anaerobic energy contribution, and of course the importance of efficiency in transferring that energy consumption into useful work. All these physiological parameters are underpinned by what is effectively systems physiology, the basic fundamental principles of exercise physiology. And the particular focus we've had for the last 10 years or so has really been looking at the importance of aerobic parameters. So in particular, the capillary supply to the skeletal muscle and the mitochondrial content and function of those mitochondria. So they're the main focal points of the talks that we're going to be around today. But in terms of understanding the role of these determinants, what we really need to have an appreciation of what are the signals for adaptation in response to training. And this is a complex area, but this slide is a very simplified viewpoint of those signals that occur within the skeletal muscle and the endothelial cells that regulate the adaptive response. And fundamentally, the exercise stressor itself, so the role of exercise itself, is a physiological stressor. So that might be the flow of blood through the capillaries, or the change in the oxygenation state of the skeletal muscle, and of course the energetic response to exercise. All these are physiological stressors which effectively switch on a cascade of signals that are sensed in the skeletal muscle cell or on the endothelial cell that switch on a series of signals, molecular signals, that contribute to the adaptive response. And then in this particular case, capillary growth and mitochondrial biogenesis. They're interrelated, so they feed off each other and relate to each other in many respects. But we really get an understanding of these mechanisms, so that if we understand the mechanisms, we can identify interventions that might target particular stressors and enhance those particular responses. So that's a very short whistle-stop tour of the mechanisms of adaptation. But the story that we came from when we started this work in endurance performance is the sort of age-old adage of what if you're already well trained? And there's a body of work going back decades and 50 odd years that demonstrates that, or suggests that, if you are already well trained, simply increasing the training volume doesn't necessarily translate into enhanced adaptation. So well-trained individuals are accustomed to performing high volumes of training, 20, 24, 25 hours a week of exercise training. If you simply increase that volume, that doesn't necessarily translate to further adaptation. So eliciting adaptations within this population is very challenging indeed. And there's really nice evidence to suggest that that's because of what we would call a reduced plasticity within the skeletal muscle in the trained state. So the ability of the skeletal muscle and the adaptive responses within the muscle are attenuated and they are reduced. So getting those adaptive responses further is very difficult indeed. And that's been demonstrated in a number of studies conducted over the last 10, 15 years or so that really demonstrates that these signals for adaptation are effectively attenuated in the trained situation. So a couple of studies here, and you can see in this one here, this is an initial signal for adaptation, the phosphorylation of AMPK, so that initial trigger for the adaptive response for mitochondrial biogenesis. And in the trained situation, or in trained individuals, in response to a standardized bout of exercise, you can see that AMPK is effectively not activated. So the activation of AMPK is attenuated. Similar for other markers of the adaptive response, so PGC1 alpha protein content in response to I think it was a 40-day training intervention, the activation or the translocation of PGC1 alpha is attenuated. And this is also reported in the signaling response, so the gene response to exercise. You can see quite clearly this attenuation of the adaptive response to acute bouts of exercise. And even when it's reported over in a time course approach, so multiple measurements of the adaptive response over the course of 2 weeks of exercise training, you can see that over the course of those 2 weeks, the adaptive response is gradually attenuating over time. So those signals for adaptation are dampened over the course of a training period. So the question we were asking, and this really started off on the back of some funding from the English Institute of Sport about, I think it was about 10 years ago now, really sort of, developed by Steve Ingham when he was the director of performance here. So a matched funded opportunity from the EIS. And what Steve was asking is, are there ways of enhancing the adaptive response to individuals who are already highly trained? And the adage that we used was, can you get more bang for your buck from exercise? So the idea of that was this is the dampened response. The hypothesis was, can we enhance that adaptive response over the course of periods of training? So these are the signals in response to exercise. And then in the red line, can we enhance those signals in response to training to potentially enhance the adaptive changes within the skeletal muscle and hopefully translate that to performance. Now, of course, I'm acutely aware that athletes and coaches have been doing this for centuries, and that is the nature of manipulating the training training theory and training principles. So the training intensity distribution has been monitored and manipulated for centuries by coaches. So I'm fully appreciative of that. We're not trying to change culture in that sense. And over the course of a small macro cycle or a micro cycle, there would be a period of low intensity training, and then you would mix in high intensity training and so on. I'm fully aware of that. What we were wanting to do was just to, are there any subtle novel interventions that might just provide that extra percentile to, enhance that small percentiles that we are wanting in that training adaptive response. So on the back of a series of work that we did, we thought about long and hard about what we could do, and we came up with the ridiculous idea of blood flow restriction. It wasn't so ridiculous because the evidence in the literature was clear from musculoskeletal physiology. 10, 15 years ago, there was a large body of work incorporating blood flow restricted exercise into low intensity resistance training. Can you enhance the adaptive responses to low intensity resistance training to get the same gains in muscle hypertrophy that you might from high intensity interval training? And clearly that's moved on quite substantially now, and blood flow restricted exercise has really gained traction, particularly in clinical musculoskeletal rehabilitation. When I gave a presentation like this 10, 12 years ago, the physiotherapists just laughed at me. They felt it was ridiculous. But now the physiologists are incorporating it into MSK rehabilitation. So it's got traction in that sense. But what we were wanting to do was to develop it in terms of training for endurance. And the model we chose was quite deliberate. So this was a result of the collaboration with Steve, and we recruited Connor Taylor for the PhD studentship at that time. And just to let you know, Connor has now moved on to bigger and better things, and he's now working at Ineos Grenadiers as a performance scientist and coach. But we started off with the idea of using sprint interval training combined with blood flow restricted exercise. The idea of using sprint interval training for this model was there's a good body of evidence around sprint interval training from Marty Gibala's work. So there's a lot of knowledge around the adaptive response to sprint interval training and so on. And it's quite a very easily controllable training bout. Whether athletes do it or not, we can discuss that later. But the idea is that we didn't want to compromise the training quality, in particular the intensity. So it wasn't going to be a low load endurance type exercise intervention. We wanted to keep that training quality high. And the role of blood flow restriction was to provide an additional stimulus. So there's already a big stimulus already from sprint interval exercise. The BFR was, could we just add a little bit more to it? So Connor's PhD, we did a couple of studies as part of his PhD that is combined in this paper, and we were utilizing sprint interval training. I'll just show a very brief video of it, if it works. Yeah, so this is Dr. Neil Martin. He doesn't mind me saying that, one of my good colleagues at work. And this is him doing a sprint interval exercise. This is actually his 4th sprint. Okay, so he's, I think he's still got up to about 1200 watts for that. So he's got a pretty decent engine. This is Connor Taylor in the early days. So that's our 30-second sprint. And in this particular model, the sprint is completed. And then what they do in a very impractical way, okay, this is a scientific model, they jump off the couch or jump off the bike onto the couch, and then we use these clinical cuffs. So these are used in clinics, put them around the leg, and then provide the blood flow restricted. So this is post-exercise blood flow restriction for 2 minutes with a 4-minute recovery, 4.5-minute recovery period between each sprint. So that's the exercise model that we chose. And this was part of our acute signaling response experiment. So what we showed, and I don't want to get into too much detail, these are the gene responses to an acute bout of exercise. In the open bars, that is the sprint interval exercise alone. So they did 4 repeated sprints. And in the closed bars, this is a sprint interval exercise with blood flow restriction. The amount of work that each time that they did each time was matched. So the matched work was matched very carefully. And these are various genes responsible for mitochondrial biogenesis and angiogenesis. So the growth of mitochondria and then the growth of capillaries within the skeletal muscle. And to cut a very long story short, what we showed was a greater gene response, so a greater gene transcription post-exercise for HIF1 alpha, which is a hypoxia signal, and enos, which is a shear stress signal. So both of these signals for adaptation, in particular related to angiogenesis, were enhanced in our blood flow restricted exercise bout. So that was interesting in the first case. So that kind of, wetted our whistle and allowed us to think, okay, well, something's going on potentially in the angiogenic response. And then we continued with the experiment. So what we then progressed on to, and this is a summary of 2 training studies that we did with Connor Taylor, and then a follow-up training study that we did with Emma Mitchell, looking at the actual adaptive response to 4 weeks of blood flow restricted sprint interval exercise, where we made measurements of performance and took a muscle biopsy before the training. They embarked on a progressive 2 times a week blood flow restricted intervention. So by the end of the 4-week intervention, they were performing 7 30-second sprints. And then we made the measurements of performance and a muscle biopsy post-training. So this data that I'm going to show now is a combination of these 2 studies. What we showed quite robustly in both Connor's initial study and followed up with Emma's study was the sort of interesting component to the performance measurements, where in the control group, these were relatively well-trained athletes, so VO2 max of 60 mils per kilogram. there was no change in VO2 max. And then our blood flow restricted group before and after training, we showed a fairly robust 4.5% increase in VO2 max. So that was exciting to us in the first place. Likewise, we showed almost a 4% increase in maximal aerobic power, so the power you achieve, VO2 max. So that was very interesting, and when Steve Ingham saw that, he was rather excited with his performance hat on. In terms of other measurements that we made, of course, we showed increases in peak power output, so the maximal sprint power output in both groups. That kind of figures because both groups are doing maximal sprints. But we also showed quite a nice increase in the mean power output during a 30-second sprint in the blood flow restricted group. So, you know, very sort of loosely, there may be anaerobic response to this sort of training as well, but that's a very sort of crude interpretation of what mean power output is. We also showed some increases, but there was no significant difference between the two groups in critical power measured using the power duration relationship. So a 3.5% increase in CP in the control group and a marginally but non-significantly different increase in the blood flow restricted group. So, you know, that's some performance measurements which are tentatively interesting, to say the least. At the same time as these performance measurements, clearly we were interested in the morphological changes that occur within the skeletal muscle, and having hypothesized that maybe angiogenesis would be enhanced in response to this training, there was actually no effect on the capillary supply. So no change in the capillary supply in response to our blood flow restricted exercise intervention, the 4-week intervention. Naturally, you get disappointed with results like this, but you don't dwell on it because you're professional and you're scientific in your output. But what we did do is what Emma sort of interrogated that sort of question a little bit more, and with a marker of what we would call endothelial cell proliferation. So this is a cellular marker of endothelial cell. We demonstrated a fairly robust increase in endothelial cell proliferation. So to put it very crudely, the endothelial cells are thinking about adapting. So those signaling mechanisms have switched on, but they haven't actually translated into capillary growth yet. So the idea is that there is still some angiogenic potential, but it hasn't translated into angiogenesis after 4 weeks of training. And there's some good arguments around that, in that Lassie Gilliman's sort of hypothesis, or Ulva Helston's hypothesis, is that, well, really, muscle capillarization is very much driven by low intensity, long duration periods of exercise, where there's a huge volume of shear stress through those capillaries. But we might be challenging that in the next presentation. In terms of mitochondrial biogenesis, again, we measured some markers of mitochondrial content in response to this training and showed no adaptive response at all. So, you know, there was no change in mitochondrial content of markers associated with that. And again, that fits with David Bishop's argument that overall training volume appears to be the important determinant of mitochondrial content, whereas training intensity, the intensity of exercise, is more pertinent to adaptive responses to the function of the mitochondria. So we didn't make any measurements of function of mitochondria in that study, but again, that leads us nicely to the next study that Don's going to present. So in relation to the argument about content and function, these two parameters of mitochondrial biogenesis, hopefully Don is going to talk to us in a few minutes in terms of new data from a 6-week training study looking at mitochondrial function. Having said that, in terms of dealing with the aerobic adaptations that I've dealt with, there's a really nice body of work from Danny Christensen's group in Copenhagen. He did a really nice set of studies using a slightly different training model that has shown really robust adaptations with blood flow restricted exercise to the other side of the coin, and those anaerobic fatigue related mechanisms, particularly in relation to potassium regulation, very important for fatigue resistance and anaerobic capacity, and has shown quite robust changes in the regulation of potassium in response to blood flow restricted exercise. So a very important sort of paradigm in relation to that we can't really forget about and needs further investigation. So having geared up Don's presentation, I think the next question that we've been asking is, well, what's the traction for athletes? And then that'll lead us nicely into the study with Paddy and the SDC colleagues who will talk to us about that. And I just want to sort of finish my little section on although much of our work has been involved with sprint interval training, we are clearly aware that there are other ways of implementing blood flow restricted exercise. So I'm not advocating sprint interval training per se, but there are other modalities that you can incorporate blood flow restricted exercise. And this is a nice review from Chris Pignanelli and Jamie Burr that really summarised that. You can use it post-exercise, during moderate intensity exercise, and different cyclical BFR. So there's lots of ways of implementing blood flow restricted exercise.